be seeing my screen, is that right?
Yep.
So thanks, Ron, so much for that very nice introduction. I'm really thrilled and honored that my colleagues in ISCB have chosen me for this award this year and given me the chance to give you this talk. And so I thought I would talk, given the context, I thought I would talk about a whole bunch of different things I've done with my students and postdocs and other lab members over the years. And the theme that kind of ties these together, which is where the title of my talk comes from, is that the work we've done has been what I call a virtuous cycle driven by technology. So we've always kept our finger on the pulse of the genomics world, the biology world, and the technology developments in that world have led to demands for new kinds of experiments, which in turn have led to the need for new computational tools, new algorithms, which sometimes we were fortunate to develop. And these in turn let people do more experiments, which led to yet further advances in the genome sequencing technology. So I'm going to tell you sort of 3 vignettes for about 12 or 13 minutes each. First, I'll talk about gene finding and past and up to the present. I'm going to tell you about some of our work in genome sequence alignment and metagenomics, which I've grouped together. And then finally, I'll talk about some of our work in genome assembly and genomic data science, which is kind of overlaps a lot with assembly. And I just wanted to mention here at the beginning that this work has taken place in sort of 3 places, not that the 3 categories of work were divided in these 3 places. First was Tiger, which you heard Ron just mention. I used to work there. Tiger doesn't exist anymore, but I was there from 1997 to 2005. It was a great, exciting place to be. At the time, it was one of the world's leading genome sequencing centers, when there weren't that many genome sequencing centers, with a special focus on bacterial genomes and plant genomes. That's one of our buildings from back in that time. And then I moved to U Maryland for 6 years, where I was fortunate to have some really terrific students whose work I'll be describing in the next half hour. And then since 2011, I've been at Hopkins, and you can see there's a picture of our campus here. So, so first, let me talk about gene finding as we used to do it and as we're doing it now. So I wanted to put up this quote from the original Human Genome Project report first. So this is this document that I'm showing here is a cover report that was planning out the first 5 years of the Human Genome Project in the early 90s. And it stated, as other documents did too, what the goal of the Human Genome Project was, which was not just to determine the actual sequence of DNA, but also, as I highlighted here, determining the location of the estimated 100,000 human genes. So we now know that that was a gross overestimate. By the way, when they said genes then, they meant protein coding genes. So we now know there's much closer to 20,000 protein coding genes, but in those days we thought there were 100,000. So that was one of the 2 major goals of the project. And so that was one of the reasons why I got interested in gene finding back in the early 90s. And I was trained in computer science, and I was looking for ways that I might bring some of the algorithms I knew about to this problem so we could have an impact. So the first result I want to talk about, not the very first thing I worked on, but the first result that I wanted to mention was a bacterial gene finder called Glimmer that we developed and first published way back in 1998. So Glimmer is for finding genes in bacteria or other prokaryotes or viruses. And it's very accurate. It can find about up to 99% of all the protein coding genes on a newly sequenced bacterial genome. And the key idea here was an idea called interpolated Markov models, also called variable order Markov models. And this is an idea that I borrowed from the natural language processing field. I had colleagues working in natural language at the time, and they were using these kinds of statistical models to model language, to model this sort of series of words that occur in a text and use that to be able to understand language. And I thought, well, if it works for a series of words, maybe it will work for a series of DNA bases. And it does. It turns out it works very well. That equation here is just is the equation from the original Glimmer paper describing the model that we used, which I'm not going to try to explain. The paper is very readable if you want to look at it. So the first 2 papers on Glimmer were published back in 1998 and 1999. That was, development was mostly done, most of the actual code writing was done by my colleague Art Delter, who was a close colleague and very long-term collaborator of mine, who's now retired. There's Art. And this program is still used today because so as I said, I was interested in human gene finding, and that was one of my motivating examples to getting into the field. So of course we didn't just work on bacteria. Eukaryotic gene finding is a lot harder. In the human genome, as you probably all know, the number of protein coding genes is about 20,000. The actual amount of real estate that they take up in the genome is around 1.5 to 2% of the total DNA. In bacteria, protein coding genes take up about 90% of the genome. So that's why bacteria are pretty easy to find genes in. And humans, much harder, and all other plants and animals, much harder. So the sort of leading model that we used, and actually other groups used as well back in the late 90s, early 2000s, was hidden markup models. Another technique we borrowed from the computer science and engineering world, and which is kind of a theme I want to keep returning to. And HMMs were used with great success in the early speech processing programs. I actually don't know if they're still the state of the art, but they were the state of the art in the 1990s for doing speech processing. And again, I had colleagues who used them, and that's how I learned about them. So we developed HMMs for eukaryotic gene finding, and one of the first ones we developed was called GlimmerHMM, and that was the work of Ella Pertea, as shown here, who was a grad student in my lab way back in the late 1990s. And we later extended to more sophisticated models, including pair HMMs. So then going on beyond that, even though HMMs were the state of the art, they weren't nearly as accurate for human gene finding or plant and animal gene finding as bacterial gene finders were. So we were looking for other ways to boost our accuracy. And one of the strategies we had was just combine all the evidence in a clever way with a dynamic programming method. And that was all embodied in a program called JIGSAW, which combined evidence from HMMs as well as alignments of cDNAs and ESTs. And for those of you who don't know what ESTs are, they were the 1990s version of RNA-seq data. So they were a way of sequencing a little bit of a transcript only at the 5' end usually, a 3' end rather, and you'd only get a fragment of it. So EST stands for express sequence tag. But in any case, having direct evidence of transcription was often better evidence than you could get from a de novo gene finder. So we developed JIGSAW as a combining method and a number of other methods too, and that was led by pair HMMs developed by others in my lab, including Bill Majoros, who was, this was all done at Tiger back in the early 2000s. So today, gene finding looks very different. We never really got those ab initio gene finders to be that accurate. They're never near 99% accurate the way we can be with bacteria. But then in 2008, along came RNA-seq. Now, we had nothing to do with that. It was invented by others, but RNA-seq combined with high throughput sequencing let us suddenly capture at a very large scale all the transcription that was happening in the set of cells or tissue. And then we had the opportunity since then to, instead of trying to rely on just the intrinsic properties of the sequence itself to find genes, we could align the transcripts directly back to the genome, assuming we had a genome, and that should tell you where the genes are. So this, in fact, does work really well. It's much more accurate than ab initio gene finders. If you have a genome and you're trying to annotate it, as I've done many, many times, and I'll mention some more examples in the rest of this talk, and you're trying to do the initial annotation, you'd like to get RNA-seq data. You can use the ab initio gene finders, they still work. But if you have RNA-seq data, it's much more accurate. So then the problem, instead of being one of recognizing statistical properties of genes and looking at a genome and finding those genes, the problem becomes one of taking the alignments of RNA-seq data and assembling those alignments with a special purpose transcriptome assembler. And the first one, I believe it's fair to say the first one to do that was a program called cufflinks. And that was developed by another former student of mine, Cole Trapnell, back in 2010, with subsequent improvements later. Cole now has his own lab at University of Washington. And then a few years later, Ella Pertea, who's now a faculty colleague of mine here in the biomedical engineering department, she developed string-tie, which uses a different algorithm and solves the same problem, slightly more accurate than cufflinks and much, much faster. I don't have time to really go into the details of any of these. I could spend half an hour just on these algorithms, but I'll refer you to the papers instead. So that's gene finding today. So we're much better at it if we have enough RNA-seq data. So, fairly recently, we decided to revisit this question that had motivated the whole Human Genome Project and has motivated me for many years, which is, what are all the genes in the human genome? Do we have an answer yet? And actually, we don't have an answer yet. So we decided to take a very large collection of RNA-seq data, GTEx, that some of you know about. We took almost 10,000 GTEx samples. There's actually more now. We took almost 10,000 GTEx RNA-seq experiments, all of them deep sequencing with about 91 million reads per sample. We aligned all of them to the genome. We assembled all of those using string tie, and we did lots and lots of filtering to try to get a picture of what were all the genes in the genome. And we had to do a lot of filtering because when you get this much transcript data, even if you don't get this much, but we've learned since RNA sequence invented, there's actually a lot of transcriptional noise. So in contrast to what we believed to be true back in the 1990s, which was back then we thought if something was transcribed, it's a gene. So now we know, having looked at lots and lots of RNA-seq data, that you can't be that quick to jump to that conclusion. So we assembled millions and millions of transcripts and filtered them down to 323,000 that we consider to be probably genes. We found a few additional protein coding genes. We're actually still sifting through those. We're not sure they're all real, but we think that maybe a couple hundred additional protein coding genes, but that number is still around 20,000. The biggest sort of additional bolus of findings here is over 100,000 novel transcripts that we found that are not in either RefSeq or GenCode. The RefSeq and GenCode for years have been the standard databases of human genes. They're independent of each other. So we kind of merged them here, and compared to either of them, we found many, many more transcripts. And that shouldn't be surprising because we started with a very large collection of very deep RNA-seq data. So of course it revealed, at least we thought, of course it reveals lots of transcripts that people haven't cataloged before, at least these databases haven't captured. And this is the work of a bunch of different people in my lab, including Ella Bertea and Elena Shumate, Gio Bertea, Alice Farabio, and Florian Beitwieser. Ella, as I said before, has got her own independent lab, and the others are members of my lab. So just one more little note on that, to sort of answer the question you might have is like, why do we even bother to do this? It's already RefSeq and GenCode. Well, when we compare RefSeq and GenCode to each other and to chess, we get a Venn diagram like this. What I'm showing here is transcripts that agree exactly between each pair of databases. So if a transcript has all of the same introns as another transcript, so the intron boundaries are precisely the same, we consider the two transcripts to be identical. And we found, to our surprise, that fewer than half of the transcripts between RefSeq and GenCode agreed with each other. And as you see here, chess actually has more transcripts in common with RefSeq than GenCode does, and it has more transcripts in common with GenCode than RefSeq does. But all three of the databases have quite a few tens of thousands of transcripts that neither of the others have. So we still haven't really figured this out. We don't have a complete list of human genes yet, and I think we'll be working on this for a number of years to come. All right, so that's gene finding today. So we've gotten better at it, but we haven't solved it yet. It's still a great problem to work on. Now I want to talk about alignment, starting with alignment of genomes and then talking about alignment of short reads and metagenomes. So this figure that I'm showing here is from a paper that described the original sequencing of the Arabidopsis thaliana genome, the model plant, published back in 2000. I was on that paper, as were hundreds of others. It was a big international collaboration to sequence this genome. It's not very big, it's about 130 megabases, but back then that was a gigantic project. And what this shows you is the 5 chromosomes of Arabidopsis. It only has 5 chromosomes. And the colored bands are showing you regions that are syntentic between the different chromosomes, where you have, in these colored regions, you have long runs of proteins that are basically paralogs. They're very similar to one another. And we, in scrutinizing this, we realized, as part of the analysis of this genome, that all of these regions that were duplicated were only duplicated in 2 copies. There were never 3 copies or more than 3 copies. And in fact, the duplications cover more than 60% of the genome, which made us realize the entire genome had undergone a quite recent whole genome duplication. And that was one of the major findings of the paper, one of the major new findings of the paper, which also goes to show you that alignment can lead to discoveries about evolution, which I've always thought was a fun thing to do. So the way we did that alignment was with another tool that we developed in my lab. That's called Glimmer. Some of you might have heard of it. It's still in use today, quite wide use. So here we took another idea. So here's this theme again. We took an idea from computer science and applied it to a problem in genomics. So this problem came up. So this Glimmer was originally designed to align 2 whole genomes to each other. It does more than that now, but that was the original idea. And the reason that we decided to do this was back when I was working at Tiger, we had just gotten funding to sequence the tuberculosis genome. TB is about 4.4 megabases. And the Sanger Center, which is now the Sanger Institute, but it's called Sanger Center at the time, had just finished sequencing the common laboratory strain of TB. And we had money to sequence a new clinical isolate strain. So this was going to be the first time that 2 strains of the same species that had ever been sequenced. And we knew that the paper, unlike all the other genome papers, wouldn't be just about the genome. It had to be a comparison between them. So I looked around and discovered at that time in the late 90s, there wasn't any software to align two whole genomes to each other. And 4.4 megabases was big. So we thought, well, we have to figure out how to do that. BLAST could not handle it, not at the time. And by the way, today there's versions of BLAST that can handle it, but not at the time. So suffix trees were something that my computer science colleagues in the area of string matching had developed many years before. And the suffix trees are a data structure, and that comes with some algorithms. And the data structure is linear in space, so the amount of room it needs, the amount of space it needs is linear in the size of the input string or the genome. And the time it takes to match a string to it or a sequence to it is also linear. So we have linear algorithms and linear space requirements. So I thought that would be good for these very large genomes. So we started with the idea that we would just find all the maximal exact matches, which turns out to be easy to do in a suffix tree. And it also pretty easy to require that they be unique. So that's where the name Mummer comes from. It finds maximal unique matches. But they don't have to be unique. You can disable that if you want. And so this little cartoon here shows you on the top, we have genomes A and B, and we first would find all these exact matches shown as colored rectangles. And then we would string together a series of rectangles that were consistent between the two and try to extend the alignments in between them. Now, the pure suffix tree part doesn't do the extension. That's done by additional code, which is part of the program called Nucmer, which is part of the Mummer package. So the original Mummer was mostly written by Art Delcher, who was a real expert in suffix trees. And then Nucmer and some of the additional stuff was written by Adam Philippi, who some of you might know. He now has his own lab at NHGRI. But Adam did this work when he was a mere summer intern at Tiger, probably about 20 years ago. So let me skip forward a little bit. So that was a whole genome alignment. A few years later, when next-gen sequencing came out, we had a new need that arose. And this was where sequencing technology is driving the computational people to come up with something new. So the selection machine came out in 2007. It was generating millions of reads per run. It's now called Illumina, of course. Millions of reads was an enormous amount of data. Now it's billions of reads, but believe me, in 2007, millions of reads was something we had a lot of trouble handling. We, of course, had many alignment programs. Alignment is one of the oldest programs in the field. There are lots of problems in the field. There are lots of solutions to it, but none of them are really fast enough to handle the output of this machine. You couldn't keep up. So it would take longer to align the reads than it took to generate them, quite a bit longer, actually. So that didn't seem like a good thing. So I was fortunate to have a really talented grad student at the time, Ben Langmead. And I said, you know, we should look at this problem, and we know about these things called suffix trees. Maybe they would be good. They're linear in time and space. Maybe you could think about adapting some of the Mummer code to do short read alignment. And he did look at that. That never appeared anywhere, but because he had a better idea, and this was totally Ben's idea. The idea was to use another technique borrowed again from computer science, from the field of data compression, the Burroughs-Wheeler transform, which is used in compression algorithms. And it does this weird thing, and I'm going to show you in this one slide sort of what it does without telling you the algorithms, because that would take another 30 or 40 minutes. But you take a string, so here's a toy sequence, 6 characters, ACAACG, $. The $sign just means that's the end of the string. You then create all circular permutations of the string. That is, you take the first letter, you put it on the end, you take the first letter, you put it on the end, you keep doing that. You generate all of these circular permutations, and then you sort that list, giving you this matrix. This is called the Burroughs-Wheeler matrix. And then you extract simply the last column from that matrix. And that last column, where I've put a little arrow here, that's the Burroughs-Wheeler transform. So you get this string GC $AAAC. So that's it. So you now have a new string. It's the same size as the original string, so it's not very big. And it might seem like I've done something that's not very useful, but it turns out this is a reversible operation. And also, there are algorithms that let you find any query string in the Burroughs-Wheeler transform in time which is linear in the length of the query. So if your query is a really short read, the time it takes to align it is a function of the length of that read, regardless of the length of the text of the genome, as long as you can fit the whole genome into memory. So it has to be in RAM. So Ben added much more to this algorithm, turned it into an alignment algorithm, which takes a lot of extra work and engineering. And the first version of Bowtie, which came out way back in 2008, was 40 times faster than its competition at the time. It got very rapidly adopted. It only needed 2.3 gigabytes of RAM, so you could run it on pretty much any desktop computer, even laptops. And then this shows you the first paper and the second paper, Bowtie 2, which was published back in 2012. And these have been just phenomenally successful and both have been cited well over 10,000 times. I should say that Ben now has his own lab here at Hopkins. He's an associate professor of computer science. So then moving on from that alignment problem, we quickly started to deal with RNA-seq data, which came out RNA-seq was invented only about a year after the selection machine came out. So suddenly we had these huge piles of very short reads that we needed to align to the genome, but they were from RNA, not from DNA. So the Bowtie program doesn't solve this problem. Now this is the problem of spliced alignment, where you have a read that may align to 2 or more locations with a big gap. And you don't want to penalize that gap because that gap represents an intron. So just shown by this cartoon here. So we'll have a little short read, the yellow guy, and it might span this intron, which typically in humans starts with GT and ends with AG, but you don't really know much more about it than that. And we wanted to do this in a way which didn't rely on annotation. You could, of course, say, if you know where all the introns are, you could just concatenate the exons computationally, align to that. And that's certainly one way to do it. But the goal of TopHat was to be annotation agnostic, to do alignment against the genome without knowing where the introns were, so that you could discover new introns. Because we figured, and I think we were correct, that there were many splicing events, splice junctions that were not yet annotated, even in human, and certainly in other species, the problem is greater. So it used Bowtie for the initial alignment to be fast, and then added a bunch of other steps to kind of stitch things together, and that was the TopHat program, which was led by Cole Trapnell, a former grad student at the time. And so we kept working on this. As nice as it was, TopHat wasn't as fast as we wanted it to be, and there was a lot of competition in this space. So a few years later, another grad student, Daewon Kim, I should say, was also the lead on the TopHat 2 program, which is a successor to TopHat. He came up with a brand new idea to make things quite a lot faster, and that is to use hierarchical indexing. And he implemented that in a program called HISAT, where the HI stands for hierarchical indexing. And the idea of a hierarchical index is you still use the Burroughs-Wheeler transform, still using this data structure. And you have one for the whole genome like before, but once you've anchored part of this little tiny read, you now know roughly where it goes. So it'd be nice if you could look at a much smaller index to find where the rest of the read goes. So he created indexes that were only indexing 64 kilobases of DNA and had them tile the whole genome with a little overlap between them, which requires about 48,000 index files, which initially seemed to me like it was going to blow up memory, but actually with some very clever development and engineering, Daewon was able to fit the entire thing into less than 4.5 gigs of RAM. The result was a spliced alignment that was 50 times faster than TopHat 2 and just as accurate. And he published that back in 2015 as HISAT, and just a year ago published HISAT 2 and HISAT genotype, which I won't really have time to talk about. And Daewon is now has his own lab at UT Southwestern. He was a grad student and a postdoc when he was doing this work. All right, the last alignment topic is metagenomics, which you might think is not an alignment problem. Some parts of metagenomics are and some aren't. So metagenomics, just quickly, you probably all know this, but I'll quickly just sort of say what I mean by that. I mean, you have, using the power of sequencing, we go out, look at an environment, it might be the human body, it might be soil or seawater, and we sequence everything there. And then we have to figure out what it is, what we got. So we get a huge pile of millions of reads, and the first question you typically would ask is, what's in my sample? So that is, you can look at that as an alignment problem, because you could take each read and you could align it to everything ever sequenced, say with BLAST, and that would be one way to answer that question. So even though I'm categorizing this under alignment, the program we came up with to solve this problem is called Kraken, and Kraken is alignment free. We're going to identify reads in a sample, so it does classification of reads in a metagenomic sample. That's all it does. It tries to put a species label on every single read if it can. And it does that by identifying short, exact matches of length K, or Kmer matches. It is super, super fast, more than 200 times faster than MegaBLAST, which is the fast version of BLAST. And we published that back in 2014. I'm going to give you like one quick slide describing kind of how it works. And this was the PhD work of Derek Wood, another former student of mine. So the idea is we're going to take all the genomes you want to be able to recognize, could be everything, could be all bacteria, and we're going to, this is not shown in this slide, but we're going to pre-process them. And for every Kmer, let's say K is 31, for every Kmer, we're just going to store that Kmer with the identifier of the species that we saw it in. So every species has a taxonomy identifier or a taxon ID. So that Kmer is just going to have one thing associated with it, one ID. And now some Kmers, of course, are going to occur in 2 or more species. So for those Kmers, we'll still store them, but the ID we'll store will be lowest common ancestor of all the species that we've seen that Kmmer in. So now we've got this database, which is really just a table with 2 columns in it, very simple. And now when we get a read, so here's a read, we're just going to march through that read 1 Kmmer at a time, and we're going to look up each Kmmer in our database and pull out the identifier of what that Kmmer looks like. Most of the time, all the Kmers are going to agree with each other. They'll all be the same species. They'll all be perhaps in the same path in the taxonomy tree. So in this example, it's slightly more complicated. We have some Kmers that are high up in the tree, some are further down. We look at what parts of the tree were hit. We make a little subtree here, and we say, oh, well, I see that there are 14 or 15 reads going down this left-hand path. So what Kraken spits out is the leaf node of the path that has the most Kmers attached to it. And it's very fast. And the reason you can get away without doing alignment, and this is critical to this task, is we define the task as simply identifying the species. So I don't really care where the read came from in the genome. I just want to know what the name is of it. What's the name of that genome? So we don't really have to worry about doing actual alignment. As long as the Kmers are long enough to be uniquely identifying, it'll give us the right answer. And generally it is very accurate. We've gone one step beyond that fairly recently in 2019. We developed, this is the work of Florian Breitfieser, a former postdoc. We have something called KrakenUniq, which doesn't just count how many reads went to each species, but it also counts without any real penalty, it's just as fast, counts how many Kmers you saw in each species. And the reason we need that is very specific to one application that I'll mention here and maybe at the very end, which is we've sometimes, an interest of mine is to use metagenomic sequencing to diagnose infections. So that's a particular problem where you get a biopsy from a person, you get millions of reads, most of them being human. And some of them might be from a bacterium or a virus or a parasite that's infected that person. We want to use sequencing as a diagnostic assay. And the problem there is that because the sample is dominated by human, you can get reads that are aligning to a bacterium when that bacterium is not really present. It's a piece of human, but it has some low complexity sequence in it, say a run of all A's or just AT, AT, AT. That may also by chance occur in a few bacteria, and then you get a false positive. So what that looks like over here on the left, I have this little cartoon showing a little green genome, a little purple one, and I might have 5 reads that hit the green genome, and they all hit the same place. 5 reads may hit the purple genome, they all hit different places. So if you ask me whether the purple genome was present in my sample, I'd say, yeah, probably it was. But if you ask me about the green one, I'd say, no, probably not. Because if it was, why would all the reads hit the same place? That's probably some low complexity region. So what we can do is we can count how many Kmers in the genome were hit. And what you'll see in genomes that are false, in reports that are false positives, you'll get the number of Kmers that won't be very large. They'll all be, because they're all the same location. So KrakenUniq gives you that without really any penalty. Okay, so that's part 2. So now I'm going to move on to my third vignette, which is assembly and genomic data science, which I'm kind of all going to cram into one 13-minute segment. So genome assembly is a very big problem. I've been working on this since my days at Tiger 20 years ago. So I'm just going to talk about a few things we've done, developing assemblers. A lot of our work, and almost all of our work in the last 10 years has been developing assembly methods that are in response to novel types of sequencing technology. But a lot of this started back at UMaryland before we really had short reads, started with long reads. And we developed a number of assemblers and then started working on various other problems like assembly forensics. Finding errors in assemblies is a big theme of some of our work. And I just want to mention these 3 guys here shown in the slide. Adam Philippi and Mike Schatz are both students at UMaryland. Mihai came with me from Tiger to Maryland as a scientific staff member and is now a full professor there, in fact, director of a big institute there. And they were sort of the key assembly team that I was very fortunate to work with and did a lot of very innovative stuff. This fourth paper I'm showing here is just the 3 of them. I wasn't involved in that paper. So I'll talk about a number of different problems we work on. So let me start with something that's not exactly assembly, but something we're aware of because we do assembly. And that is the data in the trace archive. So the trace archive was created as part of the Human Genome Project as a place to stick the Sanger reads as the sequencing was going on. Today it's called the SRA, the Short Read Archive, or Sequence Read Archive, right, excuse me. It was called Short Read Archive for a while, then they changed it to Sequence Read Archive. But anyway, back in the early mid-2000s, there was a project to sequence 12 Drosophila genomes, which was a very big project at the time. We at Tiger were sequencing a bacterium called Wolbachia, which is an endosymbiont of Drosophila melanogaster. So I knew about that biological phenomenon of endosymbiotic bacteria. And in a casual remark, Mike Eisen just casually remarked at one point that, I'll bet a bunch of those Drosophilas that are being sequenced as part of this Drosophila 12 Genomes Consortium are also have Wolbachias in them. So I thought, well, why don't we look? So we had a Wolbachia genome, we had just finished it. I could take that and use it kind of as bait and search the trace archive for all those 11 genomes and see if any of them had Wolbachias that were similar enough to our Wolbachia to match, again, using Mummer, our tool. And in fact, we found 3 entirely novel Wolbachia species. May not seem like much now, but it was pretty exciting at the time. And we wrote this nice paper back in 2005 describing those 3 new species that we discovered. And I got to use the word serendipitous in a paper title, which I'd always wanted to do. More recently, in the last 6 or 8 years, we've been looking at long read and short read assembly combined. So today we work, so our favorite recipe today for doing assembly is to generate a mixture of very long and short reads from Illumina for accuracy and long reads from Nanopore or PacBio. And a senior scientist in my lab, Alexei Zimman, has been working for many years now on an assembler called Mazurka, which has got as one of its main modules this hybrid assembly technique. And this is illustrated just here in one slide. I don't really have time to give you any details, but it basically turns your long reads, which might have a high error rate, into long reads with a low error rate by using Illumina reads. And then you can assemble those long, accurate reads together to get really spectacular assemblies compared to what we were getting just 6 or 8 years ago. So now, sort of revisiting this theme of how assembly technology, sequencing technology kind of spurs us to do things computationally. One thing we've been able to do is take on ever larger and more complex genomes as sequencing has gotten better. And it's also pushed us to improve our assembly technology. So this is a picture of the giant sequoia, which is a project we're working on right now. It's not published yet. This paper in bioRxiv, it's a preprint out. So pine trees have really big genomes. Conifers have really big genomes. So the first one we really attempted was Loblolly pine. It is a 22 gigabase genome, about 7 times bigger than human. When we first took it on, we only had Illumina technology available. So we sequenced and assembled this with Illumina only, with many different insert size libraries. This was a project that was led by my colleague David Neal at UC Davis, who's a conifer genomics expert. And he approached me with this sort of challenge. Do you think you could get this together if we could generate the data? And of course I said, sure, we'll find a way to do it. And we did. We published this in 2014. Loblolly pine was the first conifer genome that was assembled. The Contag N50 and Scaffold N50 weren't that big. Scaffold N50 was only 67 kilobases, so it was in a lot of pieces. But we did get it together. We believe, most of the genome is contained in the assembly. We had one special trick that we used here, which we used over and over again for other conifers we did since, which is that pine trees have a seed, you call it a pine nut, that is haploid. So we get a lot of DNA from one of these seeds. So for Loblolly pine, so haploid DNA is of course much easier to assemble, because there's only one copy of each chromosome and there's no differences between them as you would have for a diploid genome. So we got 60x Illumina data from a single seed here. So we employed the same strategy for a whole series of conifer genomes that we've done over the past now 9 years. Starting with Loblolly pine, we then moved on and sequenced sugar pine, which is even bigger at 31 gigabases, Douglas fir at 18 gigs. And then currently we're working on the sequoia, the giant sequoia and the redwood genomes, which are 8 and 26.5 gigabases respectively. Redwood, the paper is not even written yet, but we're using the same strategy biologically in that we always get a seed for DNA, but our sequencing strategy has moved on. Now we're using combinations of short and long reads and the Mazurka assembler I just mentioned. And one other big genome that we were able to sort of solve with this new strategy, again, with the long read technology allowing us to do so much better than we used to do, was the wheat genome. So this is a project that we were originally worked on something called Egalaps-Tausche ancestral wheat with a colleague, Jan Dvorak at UC Davis. And the reason that was being sequenced was really as a model for bread wheat, which is what we really care about because we all eat almost all of us at least eat wheat. Wheat, unfortunately, has a really big, complicated genome. It's 16 gigabases, it's hexaploid, and it's extremely repetitive. And this little plot here gives you a quick picture of the repetitive content of wheat. This just shows you a number of different genomes. It shows the Kmer uniqueness ratio, which is a concept that Mike Schatz came up with. You basically are plotting the percentage of the genome covered by k-mers of a fixed length, and the k-mers are on the x-axis. So basically, the lower you are here, the less of a genome you're covering with unique k-mers. And the bottom line here is the wheat genome. So it's the most repetitive of all the genomes we've looked at. But thanks to deep coverage from PacBio reads, which were, I can't remember the length, but they were, deep coverage, long PacBio reads, plus Illumina, deep coverage Illumina reads, we were able to assemble this and produce an N50 contig size that was more than 10 times bigger than any previous effort. And there have been multiple previous assemblies of the wheat genome prior to ours, which we published in 2017. And furthermore, our assembled genome had 1.1 gigabases more sequence than the best previous assembly. So our assembly is about 15.2 gigs. The best previous one was about 14.1. So we've actually had more DNA, and we've recently discovered that in that extra DNA, it's not just more repeat copies. There are actually lots and lots of genes there. We found over 5,700 new gene copies in that additional DNA, which we've described in a paper that is currently under review. The preprint is in bioRxiv, and the assembly work and then the annotation work is being done by Mike Allange, who's a student of Mike Schatz's, Elena Shumate is a student in my lab, and Daniela Puyo and Alexi Zimin, who are both in my lab. So now another sort of genome data science problem that we worked on and had a very sort of interesting result I wanted to share was the African pan-genome. So this is a completely unrelated to the previous project. So this was a project that I was working on with a colleague, Kathleen Barnes, who's at the University of Colorado, who was leading a project to look at the risk of asthma and allergy among people of African descent. And she had 910 individuals for whom she had whole genome data looking for genetic markers of asthma. But we had the idea that, well, these are a very diverse group. There are 20 different cohorts. They're from many different original countries scattered around Africa and the Caribbean. And they probably have a lot of DNA in them that doesn't match GRCH38, the reference genome, very well. So what if we took on the task of trying to assemble all the DNA that's in this pan-genome that's not in GRCH38? So Rachel Sherman, a grad student in my lab, took that on. And after a tremendous amount of work of assembling all the unaligned reads from all these genomes and merging them all together, we realized that there were nearly 300 megabases in the African pan-genome that was missing from GRCH38. It was much more than we expected. It's basically 10% of the genome size is present in these additional contexts. And I should say that none of the individuals in this cohort had a bigger genome. They just had lots of bits and pieces that were missing from GRCH38. So when you combine them all together, you get this very large, surprisingly large total. And we published that in Nature Genetics about a year and a half ago. And again, along the same theme, we very recently built, only published a month ago, a human genome from a single individual. And our goal here wasn't just to assemble the genome from a single individual, it was to make a reference genome. I've long thought that we shouldn't be relying on just GRCH38. Why don't we have lots of reference genomes? But here we are, 19 years after the genome was published, we're still relying on one reference genome for almost all of the human genetics work that we do. Why don't we make more? And there have certainly been thousands and thousands of humans assembled, but no one's turned them into a reference. And by reference, I mean you make it as contiguous as GRCH38 and you annotate it with all the same genes. So that's exactly what we did. We had an individual whose data is all public, part of the Personal Genomes Project, which is where everything is public and unrestricted. We have, thanks to the Genome in a Bottle Project, deep sequencing data from Illumina, Oxford Nanopore, and PacBio hi-fi technology. So we have really good data. So we assembled that data de novo, but then we scaffolded it and filled in some gaps using GRCH38. So basically, we laid all of these contigs down onto chromosomes. So in the end, we had an assembly which is somewhat more contiguous than GRCH38. And then we mapped almost all the genes from GRCH38, essentially all of them, onto it. And that's our new reference genome. And that's work by a number of students and others in the lab shown here. We've done a lot of genome assemblies over the years. I've mentioned a few of them. This slide, this doesn't show all of them, it just shows you a few more. I'm often contacted by people who are experts on a particular organism and they need help assembling it. And if it seems interesting and they seem like they're good scientists, and if I have time, then I often say yes. And so we've been fortunate to get involved in lots and lots of really interesting and exciting genome projects, a few of them shown here. So finally, as sort of a data science topic I want to kind of conclude on, if you do a lot of genomes and do genome assembly, you realize that it's still not a solved problem. And once you assemble a genome, you have to be careful that what you've got is what you thought it was. So this is a little cartoon I made showing some little babies inside some bacteria. And that's because we had this interesting finding a couple of years ago. We were doing some analysis that for an unrelated project, and we discovered this set of protein sequences. These are all annotated proteins that we found in GenBank from many different bacteria that were nearly identical. It's a very short protein, by the way. You're looking at the entire length of the protein here, but they're nearly identical. And what's really striking, if you know anything about evolution, is that these represent 6 different phyla of bacteria and 1 eukaryotic phylum. So this is an incredibly highly conserved protein. And in every case, it was annotated as just hypothetical protein, didn't have a known function. So what is this thing? Is it horizontal transfer? This has been directly transferred amongst these very distant organisms. Is it unusually conserved, but still unknown protein? So we were quite surprised, and we dug into this a little more, and we found the answer was not as exciting as either of those possibilities. It was just, this is a translation of a human centromeric repeat, a fairly ubiquitous, very high copy repeat called HSATII. That just means satellite, sat. Yeah, but it has a name. So everybody's genome has thousands and thousands of copies. There's probably tens of thousands of copies of this repeat. And we also noticed that all the genomes we found it in were draft genomes. So a draft genome means you didn't assemble it into one piece. There are a lot of different contigs. If you have a lot of different contigs, it could be that some of those contigs are not the bacterium. And then that's exactly what's going on. Those contigs were bits of human. And if you're going to find a bit of human, the most likely bit to find is something that occurs in a lot of copies of human, like this. So we decided we would turn that around and see, like, well, how big a problem is this? So it turns out it's a pretty big problem. We humans are the biggest source of contamination in genomes. And we found that over the years, thousands of spurious proteins, they're not really proteins, have snuck into genome databases. We found 3,473 microbial proteins in GenBank that are actually all translations of human repeats. And this graph just shows you from our paper from last year, shows you that almost all of them, virtually all of them, were on short contigs in draft genomes. So you have to be careful before you submit a draft genome that you haven't got bits of your own DNA in there. And if you look even more broadly, as another former postdoc just did, you can find even more contamination. So this is a program called Conterminator. Martin Steinegger, a very recent postdoc, developed this based on a fast alignment program called MM-Seq2 that he developed in his PhD work. I say very recent postdoc because he just left a few months ago to become an assistant professor at Seoul National University. And so he looked for all cases where we had near exact matches between 2 sequences that were in 2 different kingdoms, so really far apart, like, you know, bacterium hitting an animal. And if that was the case, we assumed one of them had to be contaminant, and we found there were over 2 million contaminated entries in GenBank. Most of them are small contigs in draft genomes. So just be careful when you're searching GenBank about making conclusions. So one last comment, like, why does this matter? Well, one thing is that contamination looks like horizontal gene transfer, a topic I've been working on for many years, going all the way back to 2001 when we wrote a paper shown here debunking the claim that there were bacteria that had laterally transferred their genes into the human lineage. And even more recently, a paper I wrote where I had to debunk a very similar claim that had appeared just a couple of years ago. And it also creates a problem for this task I mentioned once already, which is trying to diagnose infections using sequencing. If a bacterial genome has a bit of human in it and you're doing this diagnosis task, then you've got human DNA in this biopsy. So of course it's going to hit that bacterium when the bacterium isn't really there. So those are 2 reasons. There are others too why contamination is a problem. So I'm going to stop there. But I finally want to just thank the members of my lab currently and shown here on the left, and all of the many students, postdocs, and other scientific staff members I've had over the years, which I've shown in the middle. And last but not least, my scientific collaborators, both at Hopkins, shown on the top here, and at many other institutions around the US and around the world. It's been tremendously exciting over the past 20 years to encounter and learn from all these collaborators. I've learned a lot about biology. From some of them I've learned about computing as well. And I'm looking forward to the next decade and to doing more collaborations like the one I've already done. So I'll stop there and take questions. Thank you.
Thank you, Stephen, for an amazing talk and amazing work. created a lot of excitement. You see the chat channel was even more active than the Q&A. There were a few moments where the audio were challenging, but I didn't want to give up the video because we are a bit too impersonal, even with that in the Corona days, but nothing that human speech recognition could not overcome.
I hope so.
So let's start with a question from Ido Friedberg. What is the estimated loss of ORF detection with RNA-seq for those genes that rarely express?
The estimated loss of ORF detection? I didn't quite catch that. What's the estimated? Oh, you mean like how many ORFs you don't find in RNA-seq data? I'll answer what I think he's asking, which is after we did our assembly of all of the RNA-seq data from the 10,000 experiments from GTEx, we looked to see how many of the annotated proteins we had never seen. And the total was either 7 or 8. So there were very few protein coding genes that we didn't see in any of those samples. Link RNAs was a much bigger set, but those are their sort of status is not as definite as this for protein coding genes. So there were a few that we didn't see, but very, very few out of 20,000.
Okay, question from Tom Langaware about ancient DNA. Are your methods for decontamination useful in context?
So is the problem of contamination most extreme with ancient DNA? Well, I've done a little bit of work with ancient DNA myself, and yeah, I'd say it's actually, they have all the problems that we have and then plus more. And it kind of depends on what you're sequencing. I was involved with Hendrik Poinar, a collaboration with Hendrik Poinar, sequencing Colombian mammoth about 10 years ago now. So we had DNA from the inside the bone of a mammoth. That wasn't, contamination was mostly bacteria, but you can tell mammoth DNA from bacteria DNA pretty easily. It's a bigger problem if you're doing Neanderthal, which of course I have never worked on, but because it's so close to human, then the challenge is how do you tell the human DNA, modern human DNA from ancient DNA? So that's a bigger problem, but that's not something I really have any sort of more insight into.
Okay, a question from Yang Rao. What is your opinion about homology-based gene finding as alternative to strictly ab initio prediction now that we have so many well-annotated genomes?
Yeah, that's a great question. So I didn't really say this, but homology-based gene prediction is probably as good or better than doing RNA-seq, than having transcription. So for those who don't know, that just means if you have a new genome, you can take the DNA, you can translate it in all 6 frames, look for proteins that are similar in that organism or other organisms. And if you find a match, then you can be pretty sure that's a legitimate protein. So that also is a very important tool to have. Defining the precise exon intron boundaries is a little harder with that, but that's where RNA-seq can perhaps be a little better. But that only works if you have, so it doesn't work as well for non-coding genes, of course.
Okay, a question from Marta Lorette. What is in the 300 megabase that you find from the African pan-genome?
Yeah, so that's a great question, and we get that a lot. Of course, the reviewers for the paper wanted to know too. So the large majority of the sequence in the African pan-genome is repetitive DNA. So I think it's on the order of 75 to 80% repetitive DNA. So these are repeats that are recognizably human repeats, but they're divergent enough from GRCS38. We have the precise percentages in our paper that they're clearly different copies. Now keep in mind that the human genome itself is 50% repetitive, so it's not that much more than that. But most of it is not, most of it is repetitive DNA. There are some additional, looks like additional bits and pieces of exons and of coding genes and of non-coding genes in there. We're still working to analyze that. But we didn't, so our work is really focused on just finding the extra DNA and then figuring out later, is there any sort of, what are the functional consequences? But it's mostly repeats, novel copies of repeats.
Okay, question from Kay Nisselt, referring to your editorial in 2019 about genome annotation. You said that errors in annotation are just as prevalent as they were in the past, if not more so. Could you comment where you see we computationally focus on our improvement besides long reads?
Yeah, I know I wrote that. So there are a couple things I might have been referring to. Let me just focus on one. So the way we do genome annotation today, so we're sequencing genomes at a greater rate every year. The real annotation is figuring out what some particular protein or non-coding RNA does, what's its function, and putting a real name on it. That is still slow, painstaking, and requires usually wet lab experiments. So the vast majority of annotation that we do is just transferring assignments based on alignment from other genes. So one reason that I said the errors are just as prevalent now as they were in the past is that we're still doing that, only we're generating more DNA. So any gene, that's why I gave an example of the so-called proteins that are in now, there's now thousands of them that are in bacterial genomes that are actually translations of a human repeat. One reason that happened was that probably, I'm speculating, but probably 10, 12 years ago, someone first did it. They annotated that as a protein with a hypothetical function in a bacterial genome. And now someone else went along, sequenced a bacterial genome, compared it to all the bacterial proteins, found a nice hit to a bacterial protein. So they didn't realize it was human because they found a great hit to another bacterium, so it looked really good. So it sort of confirms the error and makes it even harder to identify, because now we've got actually whole families of these proteins that are labeled as proteins, and they're in bacteria, and they're not bacterial at all. So we have to kind of go back and clean up some of the errors that we've created in the past, and then try to figure out where they propagated to. So that's what I would say. And of course, doing better long read sequencing and direct RNA sequencing is a better way to annotate a genome. But I was referring to something else by the annotation problem there.
Okay, a question by Maxim Tsenkov. What is the value of having more than one human genome reference? Wouldn't it be simpler to append to a single reference genome?
Yeah, so this is an active debate among people doing human sequencing and alignment. Should we have lots of reference genomes, or should we just have one reference and then a catalog of all the variants? And those could be SNPs that we already have. They could be indels and other things. You could just append those to the same reference. There's also quite a few people who think we should have a graph that represents all the variants in a different, more complicated data structure, and then we could do alignment to that graph. So one way to answer that is to say all those are worthwhile directions to explore. But in my collaborations with geneticists, people doing GWAS studies, for example, I see that in the computational community, we can do lots of fancy things like make graph genomes. But in fact, their work is still going on. People are sequencing cohorts of humans looking for the genetic cause of disease. And they still, and when they're done with sequencing, the first thing they do is they align everything to GRCH38. They figure out where the SNPs and indels are. Then they look to see what genes are affected. That's the protocol. So if you want to change that, if you could give them a reference, suppose they were sequencing a cohort that had a lot of Ashkenazi Jews in it, then I would say they should use the genome we just created. And then they can simply drop in our assembly instead of GRCH38, because we have it all, it's very complete, it's all annotated. So they would see the same genes that they're already familiar with, and they could see where the SNPs fell. And I would say if we could do that for all the major human populations and maybe a lot of smaller populations, that could have a fairly immediate effect on how we do genetics. It's not that the other approaches like appending variants to one reference genome might not be ultimately the way to go, but if you want to have an impact on how we're doing genetics, making more reference genomes I think would be really powerful. I could be wrong, but that's